Hi, my name is Sally Ricketts and I work at the Kennel Club Genetics Centre, which was formerly at the Animal Health Trust near Newmarket in the UK, but is now based in the Department of Veterinary Medicine at the University of Cambridge. I also work closely with Dr. Lorna Kennedy from the University of Manchester, where I am also an honorary research fellow, and I would like to thank Lorna for providing input on the content of this talk today. So I did my PhD in canine immunogenetics with a focus on a particular part of the DLA in the dog. So thank you very much for the opportunity to talk to you about this subject today. So just a bit of background, what is the DLA? So the dog leukocyte antigen system is similarly named to the human leukocyte antigen or HLA system and is the canine version of the major histocompatibility complex, which is a tightly linked cluster of genes encoding proteins that have a major role in the immune system. So these are proteins on the cell surface. They are antigen-presenting proteins that present short protein sequences called peptides to other immune cells like T cells that then regulate the immune response to things like infection. They also have a role through this in disease susceptibility and resistance, and there's much known in the human field about the different associations of particular parts of the HLA system with disease susceptibility, including autoimmune diseases. They help the body to distinguish self from non-self peptides, and this is important in transplantation. So you've probably heard of tissue matching when individuals are compared prior to transplantation, and this is based on the MHC. It's one of the most genetically diverse regions of the genome, so there are huge numbers of variants in the genes that are involved in the antigen presentation, and that is the case in the dog as well, which I will go on to show you. Okay, so DLA genes and haplotypes. So within each subregion of the DLA, genes are generally inherited as sets because of the nature of the region. So there's tight linkage amongst some of the genes in the DLA, and obviously there's one set inherited from each parent, and these are called haplotypes. And they have codominant expression, so that means that proteins encoded by gene variants on both chromosomes are expressed. So moving on to diversity within the DLA. So when we talk about interbreed DLA diversity, so the diversity between breeds of dog, this is very high in the DLA, and there are large numbers of alleles, and this is really similar to what is seen across different human populations. So the table here shows the number of gene variants or alleles for the genes encoding the antigen presenting machinery in the class 2 region of the DLA. So for DRB1, there's around 300 alleles and around 35 and 130 for DQA and DQB. And DQA and DQB are subunits of the same molecule. So conversely, the partner of DRB, which is DRA, is not variable in the dog, and it has very limited variability in humans also. So in the dog, some alleles and haplotypes are restricted to a few breeds, and others are present across many breeds. So by contrast to this, the intra-breed, so the within breed DLA diversity is more limited. So often breeds will have 4 to 5 frequent haplotypes over about 5% frequency, with an average probably per breed of 5 to 7 haplotypes, and there may be a couple of haplotypes that are very common in each breed. And no breed has all of the known alleles or haplotypes found in the dog. And the figure here on the right shows that there can be differences between countries for the same breed. So for example, here we've got the Labrador Retrievers in the UK and then in the US. And you can see that the alleles are generally the same. So the colours represent different alleles, but the frequencies of those can be quite different. So just a bit about DLA diversity in breeds, and just to quickly touch on microsatellite markers. So obviously in genetics, microsatellite markers have been used over the decades in disease mapping and parentage testing and profiling. And they've also been looked at in the DLA region, but our currently unpublished data from the class 2 region shows that they can be really unstable and hypervariable in this region, and they aren't really linked to the functional DLA class 2 alleles. So they can't be used as a proxy to tell you about the diversity of the actual genes. So you really need to get to type the genes themselves. And DLA profiling, as I've shown you in different countries, shows that if you wanted to conserve some DLA diversity in your breed, you could possibly bring in dogs from other locations. But changing these profiles in this way might perhaps increase the frequency of a rarer haplotype, so it won't increase diversity per se, and you don't know why these alleles might be rare. They could be associated with disease susceptibility, for example. And it is known that for some breeds that do have autoimmune conditions in the breed, that prevalence can vary between countries. So how should we currently use the DLA data? Well, there's some evidence that individuals that are homozygous, so if you've inherited the same haplotype from your mother and father, they're slightly less able to respond to infection and because they have a less diverse repertoire of antigen presenting machinery. And if you have different haplotypes and a diverse repertoire, that's what you call a heterozygous advantage. And there's also evidence that in humans, if you both carry the same MHC haplotype, that you may be at an increased risk of difficulties having children. So it may be possible to avoid breeding dogs with identical DLA haplotypes, and that would obviously help to maintain diversity. But of course, it's far more important really not to breed with a dog affected with a suspected genetic disease. So probably in most cases, DLA typing would represent the last tool you would sort of consider when breeding and prioritising your health concerns, along with the characteristics you want to keep in your breed. So just to end with, it might be useful to discuss DLA and disease a bit more. So I've touched on the fact that there have been associations of the MHC regions with diseases, especially those with an autoimmune basis. So in terms of knowing what your breed's DLA diversity is, it might be useful to type haplotypes and monitor them in your breed. And it's probably useful alongside health monitoring, for example, of conditions, especially if you do have an immune-related condition in your breed. We know that DLA associations have been found to exist for many canine diseases, but it might not be the only genetic region contributing to susceptibility, and we're starting to find other regions of the genome, along with the DLA, that could be involved in these diseases. And this probably varies between breeds, so it's difficult to compare two breeds directly. So one allele could be a disease susceptibility allele in one breed and not in another, for example, because of other variants in the genome as well playing a part. So obviously, as a field, we're continuing to conduct research into a variety of autoimmune conditions in the dog and their association with the DLA. And so our knowledge of this area will only increase into the future. Okay, that's all from me. Thank you very much, and I look forward to questions and the discussions that we're going to have on the 3rd of May.